Welcome to the ASAP video interview series, Protocol Particulars. My name is Devin Snyder, and for today's episode, I have with me Ahmed Fahmy. Ahmed is a postdoctoral fellow from the University of Montreal and is part of the Desjardins lab and Team Desjardins with ASAP. Hello.
Hi, Devin.
Hi. So for today's episode, we're going to focus on your protocol mitochondrial antigen presentation in raw macrophages. But before I get into that, I was hoping that you could just briefly tell our audience a little bit more about the Team Desjardins ASAP project.
Okay, it's first of all, it's my pleasure today to talk to you and to present some of the protocols, like the work that we are doing in the lab of Michel Desjardins. Briefly, our ASAP project is on the role of mitochondrial antigen presentation in the pathogenesis of Parkinson's disease. We have actually two major contributions before we get the ASAP grant. Where we found that two main PD-related genes, PINK1 and Parkin, have been implicated in the presentation of mitochondrial antigen in mice. So we showed that these two proteins are a suppressor of autoantigens, like mitochondrial autoantigens. And the other big leap was that when we infect mice, PINK1 knockout mice, we showed that this infection triggered mitochondrial antigen presentation. I will call it MITAP from now because it's a bit long. So it triggers MITAP and leads to PD-like symptoms. So from there, we started to dig more into the mechanism, how this mitochondrial antigen presentation is happening, what are the cellular mechanisms and the pathways that are involved in this pathway to happen. And so this was the main thing in our grant, is to apply. We applied for the ASAP in order to try to discover how mitochondrial antigen presentation is happening and whether there's any other PD-related gene is involved in the mechanisms. So the pathway is start at the cell surface by like stressing the cells, whether with heat shock or LPS. So it's either through TLR4 or other mechanisms that we don't know. So we decided to follow the TLR4 signaling pathway and try to elucidate all the pathways that are involved in from the surface from the TLR4 until you get mitochondrial antigens presented on the cell surface, which will be detected by CD8 T cells. So this is basically what we are doing in the lab currently. And we have a paper that is already in bioRxiv that people can see right now. That shows that LRRK2 is implicated in the process as well, not only PINK1 and Parkin.
I think I have actually recently read that paper. So
okay, cool.
Yeah, anyone who's listening should go check it out. All right, so you guys are working a lot with the mitochondrial antigen presentation, or MITAP pathway that you mentioned, and so that's really what this protocol is focusing on. So I was hoping that you could go over what the most critical steps are for anyone else who's trying to conduct these types of assays.
Okay, so that's the thing, that the protocol, like this assay, it's a multi-step assay that involves two types of cells. So I would say that all the steps are important. You have to be very meticulous when you conduct this experiment because all the steps are important, and any step that you do without taking care of the details, you will have troubles at the end, so this is like all the steps are important, but I would say that most critical ones that I see people doing mistakes with all the time is that when you fix the cells and wash them, some people wash the cells aggressively, so they detach them all, and then at this end up by having no antigen presentation. You don't get any signal at the end of it. You say, oh, well, I have an inhibition, or what happened, and it's apparently very silly because it's really simple thing that you did and you didn't notice that you washed off all the cells when you wash them, because they detach easily from the plate. So this is one of the steps that are very important to be careful when you conduct it. Also, when you start the treatment, this is also important, like how you keep your LPS, because LPS is really sensitive to temperature and thawing freezing cycles. So at the beginning, we were keeping our LPS in -20, and it was not good, because after like 3-4 weeks, you start getting no effect, and then you troubleshoot the assay, and finally you find that it was the LPS that was not working. So that now we prepare our LPS to keep it in aliquots in -80. That lasts much longer when you do that. So this is one of the, like, some of the tips I would say. Also in the lysis, when you lyse your cells, you make sure that you have a complete lysis. When you read your plate, try to avoid air bubbles that will affect the readings. You try to avoid any debris that are in the plate by transferring the liquid into a fresh, clean plate. This will also help you getting really, at least you have good replicates. You will not get a big variation, big error bars when you do your analysis. So yeah, that's what I can say for the tips.
All right, that's perfect. Thank you so much. So you mentioned the treatment options. You started talking about LPS, but I think in the protocol you also focus on heat shock treatment and bacterial treatment. So for those types of treatments, are there any types of special considerations that people should keep in mind?
Yes, of course. So the big difference here is the kinetics. So between the different treatments, MyTab will be triggered at different time points. And it's very important to do kinetics when you do these kind of like different treatments. Now we have, even if people read the paper in the bioarchive, we have more now. We know more about the mechanism, and we know more about the triggers that can lead to induction. So it's not only heat shock now and LPS. You have also anything that can induce UPR, unfolded protein response. Anything that can trigger CGAS sting pathway can also lead to MyTab. So, but as I said, you need to make sure that you're looking at the right time point, and so it's better always to do kinetics and look at MyTab at different time point. This is one thing. The other thing, for example, if you're doing heat shock, we do heat shock at 42 degrees. So if you do 41, it might not work at all. So it's really important to stick, like, to have a good thermometer, and you stick at like you do heat shock at 42 exact. So it's really like that. As I said, like, the details are very important. So these kind of things, it matters, and it makes a big difference at the end.
Yeah, that's good to know. Okay, so people need to be really precise
Exactly.
in doing this experiment.
Yeah.
So before I let you go, I would be remiss if I didn't ask you if there was just any other advice for someone who's looking to start these types of experiments.
The only advice I would say is that to be very meticulous when you do the experiments. You have to do step by step, and you stick with the protocol. And if you get no results, you have to troubleshoot the protocol, and see which step that you didn't do it properly. And this will be the only thing that I would say, because there's nothing else. The protocol should work straightforward when you do it in the right way. So be meticulous. That's it.
All right. Thank
Yeah.
you so much, Ahmed.
Thanks a lot, Devin. Thank you so much.